I'm happy for you guys to read that. You want me to read that out loud to you? I don't think anybody wants me to do that. So I don't think Imugene needs an introduction, but I'll tell you a little bit about how we started. Roughly in about 2013, our first B-cell therapy came from the Medical University of Vienna. It was co-invented by Christoph Zielinski and Ursula Wiedemann. And we were able to parlay that science into the clinic as early as 2017. And as you know, Hervax has had some great results from phase 1B, and it's currently in phase 2. June of 2018, we then bolstered our B-cell therapy by acquiring a myriad of molecules from the from OSU and Mayo Clinic. I'm going ahead of myself. But just this Monday, we were able to announce that we are acquiring a platform technology of an oncolytic virus known as CF33 from the City of Hope, invented by Professor Yuman Fong. Yay! And you may or may not know the Imugene's team. So this, when I found out that Paul Hopper, former ex-Viralytics chair, was looking at an oncolytic virus, I quickly pulled on his jacket and said, why can't Imugene have it? And he said, no, I have other VC companies looking at this. And I said, no, we need to look at it. And so I quickly went to City of Hope, met with Dr. Fong, charmed him, hopefully, enough so that he wanted Imugene as a partner of choice. So here we are today. Dr. Axel Hughes, who quietly told me that GSK is looking for an OV, so he's going to recuse himself from the due diligence. But who I got to champion for us is Dr. Jens Eichstein. Now, this man, he's currently the managing director of Apollo, but previous to that, he was the SR1 president, and that's the venture arm of GSK. So he has seen every single oncolytic virus, every single immunotherapy out there. He's a Harvard graduate, MIT graduate, pretty smart man, and having seen every single oncolytic virus that we'll talk about today, he says you couldn't find a better virus than this one. He thinks it's better than any predecessor. He calls this the next oncolytic virus. We have Mr. Charlie Walker as a non-executive director, and Dr. Leslie Russell just is antsy about developing this molecule. She has previous experience at Amgen, Eli Lilly, Teva's, Cephalon. You couldn't get a better experienced person on the board. We have a management team that has delivered on our B-cell therapy, and Bonnie and Anthony's here with us tonight. And the executive summary, the short of it all, is that we are acquiring a platform technology of oncolytic virus known as CF33. Investment highlights, I think these are obvious. Novel technology. We are bound to be in phase 1 as early as 2020. The GMP material is already made with the PDL1. The CF33 by itself as a single agent is commenced. We have robust, so this is very important. So we have IP going out to 2037. We're just starting, so we've got a long runway. And so you could imagine when a pharma company looks at this, how attractive that's going to be. We have highly experienced oncolytic virus. So oncolytic virus is relatively new in the immuno-oncology world, and to have people that have already done this, and you'll see in a later slide that some folks have done this 2 or 3 times over, and have parlayed it to Amgen, to Merck, so we've got those people coming to join Imugene. The application of this OV is that it's readily combinable to any checkpoint inhibitors out there, and perhaps small molecule, perhaps chemoradiation. The application is wide and it's robust. You couldn't get a better partner than City of Hope and Dr. Yuman Fong. The licensing agreement was quite long, but I think we were able to bend them our way, so it's quite attractive. So the excitement of oncolytic virus is real as early as 2018. If you look across all 3 of these companies, small companies that eventually partnered with bigger pharma companies, these are just upfront payments alone, and it was upwards of $1 billion just upfront payments, and that includes Viralytics that sold for Merck for $500 million just last year. And as a Viralytics, as a case study, 28 cents at 2014, sold at June 2018 for $1.75. Some of you may have sold earlier than that, so this is time to redeem yourself. So without further ado, I'm going to bring up Professor Fong, and I know Bonnie did a great job of introducing him, but I have to tell you, he's the kind of person that knows everything about everything, has lots of dealings. He knows more people at Genentech than I do, and I work there. And when his 3 daughters who are oncologists, who are just overachievers, when they call, he leaves the room and answers their call. His daughter was going into surgery, and he said, "Excuse me, I have to answer this call." I think that's the greatest. That's the greatest for me. So, without further ado, Professor Yuman Fong.
Thank you very, very much. What Leslie meant is that my daughter was going to work as a surgeon yesterday. not that she was going into surgery. Okay. Yes. And she had a great, amazing case, and she wanted some tips, and so she called me yesterday. So again, very excited to be here, and a lot has happened in the last few weeks. And but I just want to take a few steps back and just go over with you oncolytic viruses and where things are and where things have been and where we're going. Now, Mark and Darby will have to listen to this twice, okay? And so again, I thank you for coming to lunch yesterday, and let me just go through this. The whole idea of oncolytic viruses is just trying to find viruses that kill cancer, and the idea that a virus can do that is not new, and the idea that viruses can infect only particular cells is actually very universal. When you have hepatitis viruses, they only infect liver cells. When you have meningitis viruses, they only infect brain cells. So if you can find a virus in nature or design a virus genetically and do genetic engineering to perfect it, theoretically you should be able to find viruses that only infect cancer. And that's what this entire field has been trying to do. So, 1970s, there were viruses that were found in nature to kill cancer, and a lot of trials were done. But the viruses that are in nature that are really meant to infect other things were just too toxic. And so 1990s rolls around, and the whole field became new. And that's because genetic engineering became really facile. And now we can actually engineer viruses to go kill cancers and insert genes and delete genes and do many things to manipulate the virus. So 1990s is really the beginning of this entire field. But back then, we were so afraid of hurting people. And you can understand that. We were about to design something that we send in that replicates, that infects, and kills things. And so everyone was just too worried about killing patients, hurting patients, and having major side effects. So we made the viruses very, very attenuated. They were not very toxic at all. That's why there's not been a virus given to man that was genetically engineered that has actually reached something called MTD, maximal tolerated dose. We've never hit a dose where we hurt a patient enough to say, let's slow down, okay? And in any virus that's currently in trials. And that's very unusual. And if you think about all the different chemotherapies and other drugs that we've given to patients, almost always we hit the MTD at some point. And so there's no doubt we can give viruses safely, but the viruses currently in human trials, just too safe, okay? Barely infected and killed cancer. Then the government didn't help us, and that's because they were worried that clinicians and scientists were going to hurt patients. So what they said was, go and do phase 1 trials, single dose, then come back with the results, and then maybe we'll let you do multiple doses, then come back with the results, and then maybe we'll let you do combination therapies. So suddenly you're 10 years down the line and half or 2/3 of the IP, the intellectual property, the patent, is gone. That's why almost every virus currently in phase 2, 3 clinical trial, not only are they barely able to kill cancer, they are all running out of IP, that last generation, okay? And I helped with a bunch of these. And so bubbling to the top, there were a few candidates that turned out to be good, okay? And so TVEC, that's the Amgen virus that was approved for human use for melanoma in 2015. It turned out to be a pretty good virus. And there were others, but most of them now are running out of IP. What we have learned in the last 20 years is really we need more robust viruses. We need them to infect and kill and hurt a much broader spectrum of cancers. It can't just be a single cancer that you've designed a virus for anymore. Well, you should actually try to target as many cancers with the same virus as possible. We also now know signals of how the cancer kills how the virus kills cancer. Not only does it infect and destroy the cancer cell by lysis, that's just making it burst. By turning and changing the local environment, the milieu of the cancer in its location, we can actually turn on immune signals that allow the body's immune system to recognize the cancers as foreign and attack them. And if you could do that, you extend the effect of the virus tremendously. And you could also extend the effects of many other cancer agents. And so the TVEC product, a recent trial by Tony Rebus at UCLA, by giving this virus at the same time as a kind of a therapy called checkpoint inhibitors, he doubled the effect, meaning that twice as many patients had response to the checkpoint inhibitor by giving his virus ahead of time, okay, the TVEC virus. So now that we know what to do. So in 2014, when it was very clear that the TVEC product was about to be approved, a number of us said, let's restart the field. We know what to do now. Let's go make better viruses. And so that's when I packed up my bags and left New York City. My family and I had been in New York City for 30 years. I had worked at the Memorial Sloan Kettering Cancer Center, and I needed a little bit more time to think. It's also that at the City of Hope Cancer Center, it's a small research center in Los Angeles, but it believed in inventions, and it had an infrastructure to do high throughput screening. It had infrastructure to do many of the correlative science things that you need in order to develop new compounds, and it had GMP facilities. Whatever I designed in the laboratory, if it's good and I get funding, I can actually make into human grade product to give to human beings in trials within 18 months, okay? And that's why I went there. So when I went out there, I decided to do the following. I said, why don't we take vaccine strains of vaccinia viruses, and let me explain. Vaccinia viruses are the viruses that poxviruses, and the most important members of that family actually cured smallpox. These are the viruses that were given to millions of human beings in order to wipe out smallpox in the world. So it is a virus that's very well known and very well known given to man. I had a choice. That's because the two best platforms out there are herpes simplex, that's TVEC for Amgen. Those are the viruses that causes cold sores normally, or vaccinia viruses. These are the two best platforms. And I said, Amgen's out there buying all of the IP on HSV. And I knew that because I had helped bring many HSVs forward to trials, and all the IP was being bought. Okay? And therefore I said, nah, why don't we go to vaccinia, which is a very good platform. So we took 9 vaccine strains, put them into cancer cells, and allow nature to do its work of rearranging the genes. That's because I said for 20 years, we as scientists were trying to be so smart. We thought we knew what would make a virus really effective in cancer. We would delete the single gene or add the single gene, and we turned out products that were good for writing papers, but very few that actually reached man, and only one that was approved. And I said, nah, why don't we let nature do its job, because nature is really good at making new viruses. Let's go put 9 vaccine strains into the same cancer cells and let the genes rearrange. Then let's pick out hundreds of clones from this pool, and then let's put them through high throughput screen. What high throughput screen is this, is you could take cancer cell lines of many, many different types and throw virus on and see which ones are killed. And so we chose to use this NCI 60. That's a National Cancer Institute 60 cell line. There are 60 cell lines that the NCI of the United States actually uses to screen new products that are coming to market or coming to testing. So if anybody invents a treatment for cancer, they immediately throw it on this 60 cell line and see how many it would kill. Usually, if it kills 2 or 3 different types of cancer, everybody's very excited, okay? And I said, why don't we just pick hundreds of clones out and see which ones killed all of the 60 cell lines. Didn't want to make a treatment for 1 or 2 or 3 different cancers. Let's just go find those that could be potential treatments for any cancer that's out there. Then I said, why don't we go take those that killed all 60 or most of the 60 and give it to animals and see which ones are safe. That's because if you could kill every cancer and you were safe when injecting it to animals, then it might be a really good product, okay? So took a very industrial kind of approach at developing this. Then I said, let's take all of those that are safe and see which ones turn on the immune system. And turning on the immune system means turning on the signals that checkpoint inhibitors target, PD-1, PD-L1, CTLA-4, and many of these, means recruiting CD8s. Those are T cells that kill things. They kill bacteria, they kill fungus, they kill cancer. Turn down Tregs. Those are the T cells that suppress the immune system so that cancers can't be recognized by the immune system as foreign. So that's how CF33 was born, okay? This was an industrial virus that we decided to pick because it killed everything. It was very safe in animals, and it turned on the immune system. So theoretically, it should work for all of that. Kills all of the NCI60, and it's much better than parental. Better than any of the 9 that we put into the soup itself. We've sequenced it, completely unique, completely protectable. This life form never existed on Earth, okay? Shrinks tumors at very low dose in animals, okay? So not only is it safe in animals, we've given up to 10 to the 8th and 10 to the 9th doses to animals and have it be safe. It kills cancer completely in some models at 1,000 particles, at 1,000 particles. Let me put that in perspective. Most other viruses that are currently in testing are moving to man. You need millions, tens of millions, or hundreds of millions of particles to cure animals. And this one cures many models at 1,000 particles. And you could inject a tumor in one place in the body and have the metastatic disease shrink. And that's both by direct spread of virus or turning on the immune system to go recognize those areas. And now we also know we can combine it with checkpoint inhibitors. We can combine it with cells, and we've been testing T cells, CAR T cells, NK cells, and trying to figure out how those combinations work and which combinations make sense to move forward for which disease. So I have this side over here because we anticipated people will say, how does it compare to Cavitec? Okay? And we've been getting that question a lot in the last 3 days. Okay? First of all, our virus is a DNA virus. It's a double-stranded DNA virus. And it's a huge virus. It's the biggest virus in nature. And in fact, you could see it in light microscopy, a good light microscope. You can actually see vaccinia viruses. And whereas every other virus, you actually need an electron microscope to see it. The DNA portion of it allows us to manipulate the genome very well. So if we wanted to go put in extra genes, genes that make proteins that stimulate the immune system, we can do that. So that's why one of our viruses that's moving forward carries anti-PD-L1. But if we wanted to go and put IL-17 or OX40 ligand, we can actually go do all of that, okay? RNA viruses, which are like Cavitec, are much harder to go use, okay, and manipulate. And that's why Cavitec is a non-modified natural RNA virus. The only RNA virus that is, I know that's moving forward that has really good way of being manipulated really is an NDV virus that Peter Polisi and I worked on, and we licensed to Merck, but that was 6 years ago, and they keep saying they're moving it forward and see no movement. And every 6 months we call them and nothing moves, okay? But they might be. It also is at the CF33, huge range of cancers that it could kill. Again, everything in the NCI60, and we've tested many other tumors that we think have an immune signature and just to make sure we have enough targets. Very low dose, so much bigger range than Cavitec. Cavitec mainly attacks those cancers that are ICAM1 positive, and that's the mechanism by which it works. And much lower doses, huge therapeutic window for what we have. We can make it in high titers, and we know that already. And we can give multiple doses without worrying that it will not work. And let me say that again. Viruses, we always worry as we give viruses. Yes, we want to turn on the immune system so that the viruses can stimulate the immune system to recognize cancer as foreign, as a positive thing. But on the other side, many viruses are recognized by the immune system so well that giving more than 1 or 2 or 3 doses is very, very hard, okay? Because the body just wipes it out very quickly. So adenoviruses, wiped out very quickly after the first dose, and if you gave a second dose or a third dose, usually it's gone, and you worry about that for most of the common cold viruses, okay? So, just as a summary, tumor targeting, self-replicating, effective for primary and metastatic disease, minimal side effects in animals, and so again, this we have lots and lots of data. Combining it with other therapies, whether it's checkpoint inhibition or other cell therapies, we have really good data, and much that we're currently working on. Stimulates the immune system to recognize cancer. And again, now with TVEC approved, we know there's a regulatory path to approval. We now have seen it and know what the approach is, and so that's also very helpful. So again, this is a virus that's in the orthopox virus family, and in that family were the vaccines that actually cured smallpox. So again, the FDA and the public are very, very used to seeing this virus and understand that it's safe. It has a very short life cycle, and it does not integrate into the DNA of the patient. So again, shouldn't have long-lasting genetic effects on patients, and that's because this virus actually lives outside of the nucleus, and so that's also very important. Has high insertion capacity. We can actually change up to 25 kilobases in this virus and still be okay. And again, we can make high titers, does not integrate into the host genome, and could be introduced into the patient intratumorally by direct injection, intravenously, intraperitoneally, so we can also give it not only into the vein, but if somebody had disease that is distributed in the abdomen, such as ovarian cancer, gastric cancer, pancreatic cancer, a number of different cancers, we can actually concentrate the virus in its local area by putting it into the belly itself, okay? So many, many different ways that we can give it. This is just how we made it. So again, hundreds of isolates that we looked at, high throughput screening to see how it killed all these various cancers. And again, this is one of the best that we have, and that's why we're moving forward with it. This is just some safety data. Here it says 500 because, again, when we were working on this with the Paul Hopper's other company, Vaccinia, that was the number I gave him. This is now a few months later, and we've now treated over 700 mice with very few side effects, and so we know it's safe. And some of these mouse models include mice that are called NSGs. They are severely immunocompromised mice, and usually they're very sensitive to everything, okay? And we have found it to be safe all the way up to the 10 to the 8th level, even in NSGs. And again, we don't find it in the ovaries, the brains, the lungs, the liver, the kidney, the blood, but we find it in the tumors. Now, this is 2 different viruses that we created out of this backbone, okay? This just is meant to show that we think of this as a platform. This is not just a single virus we're taking forward. This is a platform onto which we can actually put different transgenes in case down the line we think producing proteins of various types in the local area will make a big difference, and that's where the field is going. This is triple negative breast cancer, triple-negative breast cancer in an animal. And this is the deadliest of the breast cancers. These are the ones that don't respond to hormonal therapy, that don't respond to Herceptin, and big need. And very recently, the Genentech product, anti-PD-L1, was just approved, okay, for use in this patient population. And when we give virus at the 10 to the 4, 10 to the third, so 1,000 or 10,000 particles, we can cure animals. And so quite excited about a possibility of partnering with checkpoint inhibitor companies to either combine it with an anti-PD-L1 or an anti-CTLA4 or many of these other checkpoint inhibitors. This is just a comparison to some of the products that are in human testing at present. This is GL-ONC1 from GeneLuff. This is the Oncovex. We had the good fortune of having worked with both of the groups that were developing those. In fact, in my lab, we've worked on myxoma, herpes, VSV, NDVs, you name the kind of virus, we probably have worked on it, okay? And so we have a lot of comparison data, and our virus is amazingly more effective than others that are currently in testing. And that's in the Petri dish. This is in an animal. Injected side, non-injected side. So we now know by injecting one area, we can actually have tumor shrink in a different area. And that partly is virus growing in the injected area, traveling to the other area, infecting and killing those cancers. But it's also that it turns on the immune system to actually recruit either NK cells, natural killer cells, T cells, or other cells that actually will attack tumor in the other areas. So that is a quick summary of where we are and why we chose this. And I'm going to turn it back over to Leslie.
Thank you. Well, he's here all night, folks, so you can ask him lots of questions, and it's hard to follow somebody like him. But one of the many reasons, every time I've heard him talk about the same slides, and this might be my 10th time hearing it, and I still get chills, and I can't believe that we have it. This partnership with City of Hope is wonderful, not only in the sense that we get great science that I get my hands on to develop, but we get a really great discount on a licensed GMP facility. This is a GMP facility that exists in a few places, and City of Hope is one of them. And we have already 2 batches. One batch is already made, and one batch has commenced. And if I was a third-party vendor, say Genentech, coming to this facility, I would have to pay upwards of $3 million. We're paying next to nothing for it, so we get already a very, very deep discount. We already have a study design in mind. So as Professor Fong said, triple-negative breast cancer, that's a place that it makes perfect sense for us to go in. Having come from Genentech, obviously I'm a Tecentric fan, and to combine with them I think would be just a pleasure to walk in and say, hey, can we get your PD-L1, because we have seen some significant preclinical results. So we're going to fastly go through this design, because what we're going to use and what the industry is ready for is what's called an adaptive design. So you see a signal, you move right to it. So once we establish a signal within the single agent, we'll quickly combine with the checkpoint inhibitor. And we have yet to have decided on which checkpoint inhibitors that we'll combine with, but we're quite excited about that prospect. IP, as we said, going out to 2037. He's published fairly recently, but we have held him back from publishing anymore, only because when he starts publishing, I know that pharma companies will come seek him. I want them to come seek us, and that's exactly what we're going to do. His folks are ready to publish, and as soon as they unleash, we'll take it to the pharma companies and show them what we have. OV frenzy is real. It's happening. Everyone is looking for the backbone product to get better. So if, say, Keytruda only improves 20-30% of lung, if you add this product, it could triple, it could double. That's the promise of adding an oncolytic virus with a checkpoint inhibitor. And I think we can get there. Just recently, just a couple of months ago, Transgene was approached by AstraZeneca for $10 million to produce 5 candidates. So we already have a platform with 2 candidates here, whereas transgene is just starting. That's at least 3 to 5 years away from actually having a drug that you can even put in a preclinical model. So we're way ahead, and obviously Viralytics was a very successful merger with Merck just last year. Along with this acquisition, we get the ex-Viralytics crew, so of which Dr. Lin Post. Now, Dr. Lin Post did do some due diligence for us, and what he told me, so he has been a part of BioVex, bought by Amgen for $1 billion, former director of Viralytics, bought for $500 million. And what he told me, he said, Yuman has such a pragmatic approach. He had an educated scientific thought to look at pragmatic efficacy as opposed to all the BS that's going on in OV right now. So he is happy to come on to help us develop our drug, along with other ex-Viralytics members who have exquisite experience in this oncolytic virus space. Oncolytic virus, as you know, is a relatively new field. To have people that have already done it once, twice, it's a great coup for us. Along with just the execution people, not just execution, operational people, we get the scientific advisory board that Dr. Yuman has either trained or has connected with over the years. So we get the brain trust of all these scientific-minded oncolytic virus experienced folks. The timetable here, 15th of July, we just announced. We will dispatch a notice of meeting to the shareholders on August, and then we'll hold a AGM in September 9th. And maybe sooner. So now, Imugene, I can't believe that I'm standing in front of this pipeline. When I joined September 2015, I had no idea that I was going to have this plethora of real company. We have this synergistic platforms in immuno-oncology. So here we are today, and thanks for Dr. Yuman Fong. Novel technology, compelling science, we've got GMP, we've got IP, robust vaccinia with all the experts coming on board. That is now Imugene. So thank you. And we want it pretty interactive. Whatever questions you have, please let us know. Interrupt us. And we've got it on video. He's done this every single time. There hasn't been a time that he hasn't killed it at every turn.
When's the video going to be released?
Well, we'll have to ask Rockpool.